name is Vanessa Fernandez. This summer I was working in the Summer Institute of Medical Ignorance. In my lab, I am researching conditional treatment mice that have been induced with fibrosis. I am testing the effects of a synthetic estrogen that is involved in the remodeling process of hypertrophy. Basically, the synthetic estrogen is believed to reverse the impact of fibrosis. So, some questions I had were, what is the mechanism of how the synthetic estrogen works on the cells? And 2, what does it mean to be conditionally treated? Unfortunately, these questions won't be answered in this video. However, my research question is, how does a synthetic estrogen affect fibrosis specifically in regards to the effects it has on the calcium-handling proteins, such as SERCA, phospholamban, and the sodium-calcium exchanger, which are major proteins in the heart muscle cells? The technique used for this is Western blotting. It is the transfer of a sample from a gel to a membrane. By doing this, it detects the relative amount of different proteins present in each sample.
Well, first, we solubilize our desired sample using urea to make a homogeneous solution. Next, we create an agarose gel and pipette our solubilized samples into the wells of the gel. This undergoes gel electrophoresis to let the protein run through the gel. Now time for the transfer. We make a transfer sandwich. No, not this type of sandwich. We use filter sheets, the gel, and the membrane, and place in the transfer apparatus. Wait 2 hours. The current will pass from cathode to anode, a negative to a positive charged electrode. Place in methanol, then water, then sodium hydroxide, water, PBS, place in primary antibody, wash 4 times with PBS, place in secondary antibody, and wash 4 times with PBS again. That was a mouthful. The antibodies are used because they help distinguish the target protein. Primary antibody attaches directly to the antigen. Secondary antibody attaches to the primary antibody with a fluorophore. Now, on to the scanning.
Voila! The process is complete. Now we analyze to determine the specific proteins present and in what amounts by the fluorescence measured. Specifically, in my experiment, I'm doing this to both a control group that is not given the synthetic estrogen and one that is. In this way, I can compare the differences between the two and determine if one is experiencing an increase in a calcium-handling protein I am probing for. As my review of this technique comes to an end, the questions I have now are, is there a similar method that can be used for other components of the body aside from proteins? The other question is there a more efficient method that does not take at least 3 days to detect amounts of different proteins? The third, although using a secondary antibody results in a signal amplification, could direct detection be used that only uses primary antibody? I hope you all learned something useful from this video. I hope to see you next time, and thank you, Simi.